Okay, welcome to this explainer. Today, we're diving into something really exciting, how a completely revolutionary sequencing technology is literally reshaping molecular biology as we know it. You know, for decades, we've relied on some pretty incredible tools to map the transcriptome, but the catch is we've always been looking at it through a filtered lens. Today, we're going to talk about how we can finally strip those filters away.
Think about it like this. Imagine if, instead of reading a translated, heavily summarized version of a famous book, you could actually hold and read the original manuscript. I mean, complete with the author's handwritten notes in the margins, the crossed-out sentences, the subtle changes in the ink. Well, that is exactly what's happening right now in genomic exploration. We're moving from the summarized version to biology's original manuscript.
And the paradigm shift here is absolutely stark. See, traditional RNA-seq forces us to break our sample apart, convert it into complementary DNA, and then amplify it with PCR. And during that whole process, we lose those vital, native biological notes. Direct RNA sequencing, on the other hand, reads the pure, unadulterated molecule. We are talking about single molecule resolution that actually retains every single modification. Here's our roadmap for today's explainer. We'll start with the direct RNA revolution, look at decoding through the nanopore, map out the epitranscriptome, talk about conquering data complexity, check out the cDNA solution, and wrap up with the future of transcriptomics research. Let's jump right in.
Part 1. The direct RNA revolution, focusing on PCR-free RNA analysis.
So, what exactly is direct RNA sequencing, or DRS? Basically, by avoiding cDNA synthesis and PCR amplification altogether, this method completely eliminates amplification bias. This means we can preserve the intricate genetic architecture and those sophisticated post-transcriptional regulatory mechanisms exactly as they exist in nature. It's a PCR-free method that sequences pristine RNA structures right from the 3' end to the 5' end.
Moving on to Part 2. Decoding through the nanopore. How do we actually read these native strands? Okay, so how does this work without amplifying anything? It's actually a physical process. First, we ligate a poly-T adapter and a motor protein directly to the RNA. Then, that RNA physically passes, or ratchets, through a microscopic biological nanopore that's embedded in a membrane. Now, as it passes through, it disrupts an ionic current flowing across that membrane. That electrical disruption acts as a unique biological signature for every single base, which lets us base call the electrical signals in real time. It's brilliant.
And here's the crucial point. Because we aren't fragmenting the sample to amplify it, we can read massive, full-length transcripts. We're talking ultra-long RNA molecules ranging from just 70 all the way to over 26,000 nucleotides in length. This gives us complete 5' to 3' structures without having to rely on error-prone bioinformatics assembly. You literally get the whole picture in one continuous read.
Next up, Part 3, mapping the epitranscriptome, where we keep those RNA modifications completely intact.
This brings us right back to those handwritten notes from our manuscript analogy. Did you know there are over 160 types of RNA modifications identified in nature? In traditional sequencing, every single one of these is lost during the cDNA conversion. We've essentially been flying blind to them.
But these are the heavy hitters. Modifications like M6A, M5C, and pseudouridine are directly responsible for controlling mRNA stability, translation efficiency, and splicing. And DRS spots them natively, simply because each modified base disrupts that nanopore's electrical current just a tiny bit differently than a standard base would.
Let me give you a real-world example. In a really foundational study on the Arabidopsis plant looking at flowering times, researchers used this exact technology to map exact M6A sequence motifs at single-molecule resolution. They were able to prove a direct link between 3' UTR methylation by a specific protein and rapid mRNA decay. It completely changed our understanding of the plant's flowering mechanics. That kind of single-molecule native evidence is exactly what DRS brings to the table.
And it's not just about chemical marks. This captures incredible structural data too. Over 90% of direct RNA reads capture complete 3' end information. This is huge because it allows for precise, per-read, poly-A tail length estimation. That's absolutely vital for investigating metabolic states, stress physiology, and gene translation dynamics, and you don't even have to run separate assays to get it.
All right, Part 4, conquering data complexity, or how we actually interpret these signals. Now you might be wondering, what's the catch? Well, getting all this pristine biological truth introduces a pretty massive technical hurdle. Translating millions of raw ionic current disruptions into an accurate map of RNA modifications requires intense, highly specialized bioinformatics horsepower. We aren't just looking for A, C, G, and U anymore. We're trying to identify the subtle electrical variations of over 160 different modifications.
Analyzing this data involves a really rigorous, multi-step pipeline. You've got live flow cell monitoring and QC, base calling raw Fast5 files to FastQ, doing signal-aware modification extraction, and then reference-guided splice alignment and isoform clustering. Building and maintaining all of that, it can quickly overwhelm standard lab computers.
So to solve this, researchers are deploying advanced machine learning. For example, the tandem mod transfer learning model leverages one-dimensional convolutional neural networks and bidirectional LSTMs trained on specific datasets. These deep learning tools can simultaneously detect multiple types of RNA modifications with truly astonishing accuracy. But again, you need serious computational expertise to run them.
Which brings us to Part 5, the cDNA solution. So what if you want these profound insights for your fundamental research, but you don't want to build a deep learning supercomputer in your lab? Based on our source material, this is where we highly recommend checking out the comprehensive Nanopore Direct RNA Sequencing service for isoforms, RNA modifications, and poly-A analysis offered by cDNA. It delivers all these complex endpoints, the full-length transcript isoforms, the native epitranscriptomic maps, and the per-read poly-A tail lengths, all in a single integrated run.
Why partner with them? Because they handle the whole shebang. They provide an end-to-end workflow strictly for research purposes. From total RNA sample receipt and stringent QC, all the way to publication-ready visual dashboards. They handle making those PCR-free 1D libraries and all the intense bioinformatics, meaning you get pure, biologically accurate data without any of the computational headaches.
Finally, Part 6, the future of transcriptomics research and how this is advancing science.
Things are moving incredibly fast right now. With recent chemistry iterations, accuracy rates are exceeding 94%, and base call accuracy is pushing past 99%. We're officially entering a golden age for investigating complex transcriptomes. We're talking high-confidence fusion gene detection, understanding alternative splicing on a deeper level, and even integrating this with CRISPR-Cas9 to monitor precise transcriptomic changes from genetic interventions.
There's a quote from the cDNA resource that I think perfectly captures the momentum here. The advancement of DRS is poised to uncover new mysteries of life, delivering deeper biological insights and enabling more precise scientific solutions. Leaving behind those old PCR and cDNA biases is absolutely critical for the future of rigorous, mechanism-driven research. It's all about getting as close to the biological truth as we possibly can.
So I want to leave you with this question. How could uncovering the hidden native layers of your RNA transform your next research project? If you're ready to stop computationally reconstructing your transcripts and want to start reading biology's original manuscript directly, it might be time to explore Nanopore Direct RNA Sequencing. Thanks so much for joining me on this explainer, and happy researching.